online on the theme Regulated Bioanalysis Research Orientation from August 1st to August 6th, 2022. So here the session, every day we are going to conduct a session by different speakers covering various topics which involves PG students and research scholars. And I feel privileged to extend my warm welcome to all of you. Now, I'd like to say about my college, our college, about the introduction. Komareddy Venkat Sadasivarao Siddhartha College of Pharmaceutical Sciences is the first private self-financed institution in Andhra Pradesh, which was established in 1994 at Vijayawada, sponsored by Siddhartha Academy of General and Technical Education, affiliated to Krishna University, offering 4-year graduate program, BPharm, 2-year postgraduate MPharm in Pharmaceutics, Pharma Analysis, and 6-year PharmD, 3-year PharmD PB programs. The college is approved by the All India Council for Technical Education and Pharmacy Council of India. The college is located at the middle of the city and has an excellent academic atmosphere, culture, and discipline with the best infrastructural facilities. Now coming to our department, the PG program in pharmaceutical analysis is approved in the academic year 2007. As a part of research orientation to the faculty and PG students, by the suggestion of our principal, we actually thought going for the research orientation to the faculty and PG students. So the Department of Pharmaceutical Analysis is organizing now 1-week online international faculty development program. The program actually emphasizes on modern bioanalytical techniques, which involves the extraction of drugs, BA/BE studies, pharmacokinetics, and applications and social outcomes. So before going to the session, I'd like to have a prayer song by Sarada Mrinalini. Over to Sarada.
Saranu Saranu Surendra Sannuta Saranu Sri Sati Vallabha Saranu Rakshasa Garva Samhara Saranu Venkatanayaka Saranu Saranu Surendra Sannuta Saranu Sri Sati Vallabha Kamala Dharudunu Kamala Mitrudu Kamala Setrudu Putrudu Kamala Darudunu Kamala Mitrudu Kamala Sentrudu Putrudu Vinnapamu Vinavaya Tirupati Vinnapamu Vinavaya Tirupati Vinnapamu Vinavaya Tirupati Venkatachalanayaka Saranu Saranu Surendra Sannuta Saranu Sri Sati Vallabha. Thank you.
Unmute, Vijaya, unmute yourself. Vijaya, unmute yourself.
Vijayalakshmi madam, unmute chesko.
Yeah.
Now I am pleased to invite our principal ma'am, Dr. A. Sunita Garu, to give a keynote address on this session.
Thank you, Vijaya. It's my pleasure to extend a cheerful welcome to you all for 1-week online international faculty development program on regulated bioanalysis research orientation being organized by Department of Pharmaceutical Analysis, KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. I accord a genial welcome to today's speaker, Dr. Nageshwar R. Pillai. We are highly honored to have you in this blissful morning. The management, Siddhartha Academy of General and Technical Education, Vijayawada, mainly focus in making the transmission of knowledge from the faculty to students for highly developing the next generation teachers as well as researchers, with the aim of equipping faculty members with required skills and knowledge. KVSR Siddhartha College of Pharmaceutical Sciences pursues the faculty development program as its primary objective. We, the members of KVSR Siddhartha College of Pharmaceutical Sciences, made strenuous efforts to upgrade the quality of the teaching profession at nationwide. So, we have been conducting series of faculty development programs for enrichment of knowledge in pharmaceutical realm. The main objective of this faculty development program to acquire in-depth knowledge and build a confidence on bioanalytical techniques. The program emphasizes on modern bioanalytical techniques which involve extraction of drugs in biological samples, bioanalytical method development, and validation by LCMS as per US FDA. Make to understand guidelines for BAB studies. For this 1 week program, we are blessed to have prominent speakers. For the inaugural speech, we have invited senior scientists from United States of America who work with US FDA as a bioanalyst. Most inspiring personality, Dr. Nageshwar Pillai, lead bioanalytical scientist, Center for Mass Spectrometry, Maryland, USA, is going to enrich us with much more advanced information on method development and validation by LCMS. Another speaker, Dr. Krishnaveni, and Professor and Head Department of Analysis, JSS College of Pharmacy, Wuti, will emphasize on sample preparation, an imperative tool for bioanalytical method development and validation. The session 3 covers bioavailability and bioequivalence studies by Mr. Srinivas Reddy, Quality Control Manager, Aurobindo Pharma Limited, Hyderabad. The session 4 focusing on application of pharmacokinetics in drug development by senior scientist Dr. Bharani S. Sogali, Bangalore. The session 5 explores on metabolite identification by senior academician Dr. Moshina Patwekar, Lickman College of Pharmacy, Gulbarga. The session ends with research and development to commercialization for the benefit of our state by Dr. K. Satyamurthy, Head ITC Limited, Guntur. With the participation of outstanding experts, we hope productive discussion would stimulate new creative ideas to translate new discoveries into better practice and application. We appreciate you being here. I thank all the eminent speakers for accepting our invitation and putting up your interest to share your knowledge with all the faculty members. I thank the management of Siddhartha Academy of General and Technical Education for the immense support to organize such beneficiary programs to all the pharma fraternity. A special appreciation to the program coordinator, Dr. Vijay Lakshmi, Associate Professor, Department of Pharmaceutical Analysis, and Organizing Committee, Dr. Rihana, Assistant Professor, Ms. Sarada Mrunalini, Assistant Professor, Ms. Monica Pratyusha, Assistant Professor, and all the teaching staff of KVSR Siddhartha College of Pharmaceutical Sciences for their enormous support to make this event a grand success. Thank you one and all for your patient listening. Thank you.
Thank you, ma'am. Thank you for your valuable words. And one more thing, you have introduced about the speakers, all the speakers. And now I want to give a brief biodata about today's speaker, Nageshwar R. Pilli.
Ma'am, you need to turn on your video, ma'am.
Yeah, sure. I know this sir, Nageshwar Pilli sir, from 2010 onwards. He's expertised in bioanalytical method development of various chemical entities, biomarkers, lipids, small molecules in different biological matrices, and bioavailability and bioequivalence studies by LCMS, UPLCMS, and HPLC. Now he is presently working at currently sir was working at lead bioanalytical scientist at Center for Mass Spectrometry, Maryland, USA. Earlier to this, Dr. Pilli worked with US FDA as a bioanalytical scientist. Dr. Pilli was also worked with various pharmaceutical organizations like Hetero, Pinmal Healthcare Limited, and contract research organizations over a decade. He has published more than 60 international and national research papers in reputed journals, which includes 2 published in JAMA, having an impact factor of 57.3 in 2021. He is actively involved in research areas of bioanalysis, method development and validation, ADME, and pharmaceutical analysis. So actually, we chosen this topic based on our MPharm PG syllabus, 201T, Modern Biological Analytical Techniques, which includes the first unit, second unit. This is for your information, dear participants. And not only that, for the research scholars, we would like to give information regarding the sample preparation in our labs in the college. As you know, academics is different from industrial situation, practically speaking. It's far difficult to do the same process in our lab. So just I requested sir to deal about this sample preparation technique in an easy way, like whatever the suggestion given by sir as per the practical session we need to conduct for bioanalysis samples in our college, in our colleges. Okay, thank you and welcome you, sir. Please go ahead, sir.
Very fortunate enough to have you, sir. Glad to see you in this platform.
Thanks, madam, both of you for introducing me to your team.
Sir, actually 1 o'clock for sir. Midnight.
It's almost 1:15, madam.
Sorry for the inconvenience made by us.
No problem. It's not at all a problem to worry. It's okay for me. That's okay. It's not a problem. So thank you, both of you, ma'am.
Thank you.
So can I share my screen right now?
Yes, sir. Dear participants, if you have any queries in between the session, you please post your questions in the chat box so that after completing the session, Dr. Rihanna will share your questions to the speaker. Thank you.
So can you guys able to see my screen right now?
Yes, sir. Good.
Okay, good. So thank you everyone for that joining today's session. So it's my pleasure to present my few things which I know about the bioanalytical method development validations. Today I'm going to talk about how some of the aspects of bioanalytical method validation and development and validation process using liquid chromatography, quantum mass spectrometry. So this is how does it look like. This is the liquid chromatography part, and this part is like mass spectrometry part. So I'm going to explain in brief about these things right now. So today my presentation outline is this one. So I'm going to discuss about the importance of the bioanalysis right now. So why we need, where we apply the bioanalysis and those kind of things. And next one is different LCMS platforms that are currently available in the market, and how to initiate the method development process when you have a drug or a molecule in our hand. So in the method development part, I'm going to discuss about the mass spectrometry, MS conditions optimizations, and LC optimization. During the LC optimization, so how do you choose the column? How do you select the mobile phase and those kind of things? And proper extraction conditions. So how do you choose like different proper extraction condition? What kind of solvents you are going to use the extraction? So this I'm going to discuss that also. And finally, a brief discussion about the method validation parameters as per the US FDA guidelines. Once we develop a method, how to validate it, and what are the acceptance criteria for that. So this is how we do the workflow of mass spectrometry. When you have a sample in our hand, so we cannot directly inject into the mass spectrometry. So we need to prepare the sample for this. And once we prepare the samples, it need to be separated by using LC part. That's especially column part in the LC. And once we separated the analyte from the endogenous components, that will be detected by the MS part. So whatever the component that component in the sense like my analyte or a drug, that was detected in the mass spectrometry, it will give you or output as a chromatographic. So let's see the bioanalysis. What is bioanalysis here? Bioanalysis is a quantitative measurement of a compound or a drug and their metabolite in biological fluids, blood, plasma, serum, urine, and different kind of tissues. So where we applied and where we need the bioanalysis part. So from everywhere, like for being a pharmaceutical scientist, bioanalysis is very much important. Everywhere we need bioanalysis work. Suppose you have formulated one drug. So if you wanted to see the bioequivalence and the availability of the systemic circulation in the drug, we need bioanalysis part. Like that preclinical studies, that means animal studies. If you are doing new discovery of any drugs, we need to have bioanalysis. Clinical trials, drug-drug interaction studies, and metabolite identification, bioavailability, bioequivalence studies, pharmacokinetics, toxicokinetics, TDM, therapeutic drug monitoring studies, and ADME studies, absorption, distribution, metabolism, excretion. So for all these studies, being everywhere in pharmacy or clinical related thing, we need to have a bioanalyst, and we need to develop a bioanalytical method for this, for any of the compound that we are going to study. So what is being a bioanalytical scientist, what we should have some goal and objective before initiating our bioanalytical method development. So first we should know what is our goal. Are you going to detect the compound just to identify the compound? That's nothing but qualitative analysis. Or are you going to determine the concentrations of the drug blood in the biological samples? That we need to determine first. And we should think about what the kind of instrumentation platforms we have in our laboratory. Do you have HPLC, or we have UV spectrophotometry, or do you have like LCM, SMS platform in our laboratory? And type of the matrix, in which type of the matrix you are going to analyze it. That means in which type of matrix you are estimating your drug of choices. And the fourth one is what is your LLVQ and ULVQ. That means lower limit of quantification. How low you are trying to detect your drug, how high you are trying to detect our drug is nothing but upper limit of quantification. So once we have these things in our mind, then we come to our expectations. What you are expecting? Are you going to develop a LCM SMS based method or HPLC based method with the shorter runtime? That's our expectation, right? All the time. So every analytical method should be shorter so that we can analyze large number of samples in a day. So whatever the peaks, chromatographic peaks that we get, it should be symmetrical peak shape, and we should not get any interference. The method should be highly sensitive. That means it should be able to detect very low concentrations of the drug in fluids, and the result should be consistent, reproducible. So these are our expectations before starting any method development. So these are the most widely used, some of the MSMS platforms in the market right now. So we have a lot of MSMS platforms, like more than 5 to 6 kind of things. The most widely and most widely using mass spectrometers are like ABSIX. This one is ABSIX 6500 QTRUP LCMS MS system. And second one is like Waters Corporation. This is called TQSS. This is called a TQXS. And third one is from Thermo Fisher Scientific. So each manufacturers, they have their own software development. ABSIX uses Enlist software, Waters uses Mastering software, and the Thermo uses Excalibur software. So these softwares are pretty similar. Once you have some idea about one of these software, so it is easy to learn the next software. So before I discuss about how to initiate the method development part and what are the different steps in the method development part, I would like to brief a little bit more about the mass spectrometry principle. So here, like, there are two types of ionization techniques mainly used in the mass spectrometry. One is like electrospray ionization, which is called ESI, and second one is the atmospheric chemical ionization, which is also called APCI. So in ESI, this is the ESI probe that we used in the mass spectrometry, and this one is like APCI probe in the sense it kind of needle or a metallic part in the mass spectrometry. So difference between this and this is like this look like a little bit bigger one, and this one is a smaller one. The functional wise, there will be a lot of difference, because here in the ESI probe, there is a conversion of liquid to gaseous phase, and but in the APCI gases to gaseous conversion here. So here there will be a chemical reaction happens because of that. This is also called as a coronary discharge needle. Because of that coronary discharge needle, there will be a chemical reaction between our drug or analyte of interest with this needle. But here there will be no chemical reaction. So most widely used ionization techniques are ESI and followed by the APCI here. So coming to this one, during the ionization part, there are like 3 different steps involved in the ionization of mass spectrometry. One is desolvate, I is like ionize, G is guide, DIG. So this part is the LC part, like think about it is a column. Solvent and the analyte, which is pumping from the LC part, will enter into the mass spectrometry source like this. Once the solvent enters in solvent that contains your analyte enters into the mass spectrometry source, because of the application of these gases, different gases like nebulizer gas and sheath gas, it forms the fine droplets like this. So this is called, these droplets will get shrink because of the application of high temperatures in the source, which is about 350 degrees centigrade and above. Because of this, that temperature, this droplet will get evaporated. So evaporated droplet, because of the application of the electric field or the different voltages, it forms different ions, either positively charged ions or the negatively charged ions. So these ions, once ions are formed, these ions will be guided to the mass, inside to the mass spectrometry. So this part we can see outside from the mass spectrometry, as I shown in the previous slide here, like this. This part, this is the source of the mass spectrometry, and this is where ionization happens. And this part is inside the mass spectrometry. We cannot see it. It is inside the mass spectrometry. Once the ions are formed, it will be guided towards the mass spectrometry analyzer section. This called it as a analyzer section. In the analyzer section, there are 3 different quadrupoles are there. One is Q1, another one is Q2, also called collision cell, and third one is the Q3, quadrupole 3. So what is the function of these Q1, Q2, and Q3 here? Let's see that. So Q1 is a quadrupole analyzer 1, which detects your parent compound. Suppose you are analyzing some A compound, which is having molecular weight of 300. So Q1 will detect that part. Once Q1 detected that part, it will pass it to the collision cell, which is also called Q2. So here collision energy will be applied. Because of the collision energy, your parent compound will get fragmented. It forms the product ions, different product ions. In the next slide, I'm going to show you how it forms the product ions. So once the product ions formed in the collision cell, it moved towards the Q3, that means quadrupole ion 3. So where those ions are detected. So Q1 detects the parent ion, Q3 detects the product ions. Q2 makes the product ions. Once all together, so these parameters will be detected by the mass spectrometry detector, and it will give the output in terms of our data. So coming to the method development initiation part. So suppose I'm not going to discuss any theoretical or principles like this. I'm just talking about how we do in our laboratory, simple way, how you can adapt in your laboratory based on my presentation. That's my intention. So once you are planning to develop a method for some kind of drug, obtain the pure sample, that means pure reference standard or working solution, working standard of that particular drug, whatever suppose you are going to develop a by LCM SMS based method for the paracetamol. Obtain paracetamol reference standard from the vendors or the pharmaceutical companies. Prepare a solution in the suitable solvents, LCM SMS based solvents like methanol, estronitrile, water, IPA, and DMSO, dimethyl sulfoxide. So preferably methanol, estronitrile, and water. If the compound is not soluble in these solvents, try to use DMSO. It is a universal solvent. It solubilizes most of the compounds. Then identify the molecular weight of the compound. So you know that literature you will get, or based on the structure you can determine the molecular weight of the compound. So you need to determine the ionization mode of that compound, which ionization mode you are going to use, either in the positive mode or the negative mode. And then what ionization source you are using for the mass spectrometry. ESI, as I explained earlier, are you going to use ESI mode or APCA mode? That's important. So how do you determine whether you are using the positive or negative mode? That's important, right? So most of the basic drug or neutral drugs will be ionized in the positive ionization mode. And most of the negative acidic drugs, those drugs are ionized in the negative ionization mode. It's not the predefined rule. It can be other way also, but as per my experience or the most of the bioanalytical methods, so this is going to happen. Is a neutral and basic drugs in the positive mode, acidic drugs in the negative mode. In positive mode, how you are going to determine it is like you have the molecular weight of 300, that's your M, plus 1, that's going to be another proton is going to be added for that. That's going to be 300 plus 1, 301 for this in positive mode. In the negative mode, it will be other way. Your 300 minus 1, that's going to be your parent compound. Yeah, that's you are going to detect in the mass spectrometry. So this is when you see, if you see this picture, there is a procedure called flow infusion method. So in this method, take your sample, whatever the compound you prepared in the solvent. So approximately it is like 1 microgram per ml solution is most widely used for most of the mass spectrometry optimizations. Take that drug in the syringe, slowly infuse into the mass spectrometer source like this, slowly. So once your compound enters into the mass spectrometry, here we have the two different kind of parameters in the mass spectrometry. One is like source parameters and another one is the compound parameters. Source parameters are like different gases and temperatures. Compound parameters are like different voltages. You need to optimize these parameters and these parameters which are suitable for your compound. So source parameters are common for so many compounds. Like if you are analyzing 3 compounds in a simultaneous analysis, in that case, so for 3 of our compounds, source parameters are common. That means cotton gas, if you have 20, that will be applied for all 3 of the compounds. But the compound dependent parameters are different for each drug. Suppose you have 3 drugs in your simultaneous analysis. Each drug will have different declustering potential, different collision energy, different kind of collision cell exist potential. So you need to play with these values to get maximum response like this. This is the mass spectrum that you are going to get. This is the compound I did recently, which is having molecular weight of 311, positive ionide mode. When I subject to mass spectrometry collision, it forms 3 major products here like this, 315, 161 and 177. So out of these 3 product ions, this is how it breaks. This part will break in the mass spectrometry and it forms this product ion. Out of these 3, 161 is showing highest response, consistency in the response. That I selected as a product ion. So this is the parent ion and 161 is a product ion. These are my values to develop a LC based method optimized from the MS conditions. So once you have the MS condition, next step is to optimize the LC parameters, HPLC or EPLC parameters. So EPLC parameters, LC parameters are mainly included selection of your mobile phase and composition of your mobile phase. What composition are you going to use it? So most widely used solvents in the mass spectrometry is organic solvent, acetonitrile, methanol, and polar solvents like water and isopropanol. And we always use the buffer to evaluate the compound. Some of the acidic buffers and some of them are basic buffers. Acidic buffers are like ammonia state, ammonium format, mainly in the concentration range of 1 to 50 millimolar. Formic acid and acetic acid in the concentration range of 0.01 to 1% range. And basic buffers like ammonia solution, ammonia hydroxide in this almost similar range. So we have a lot of buffers in like potassium dihydrogen phosphate, sodium hydroxide, those kind of buffers as well. But in the mass spectral analysis, we are not going to use those kind of buffers because those are not volatile buffers. If you use those buffers in the mass spectrometry analysis, what will happen? Those won't get evaporated properly. It forms those buffers deposit the salts in the mass spectrometry source. It causes damage to the mass spectrometry as well as for the HPLC also. That's why we are going to use most of the time these buffers only. Oddly, we have like 4 buffers in the buffers like acidic buffers and 2 to 3 basic buffers in the mass spectrometry. So how do you choose your mobile phase composition? In the reverse phase chromatography, all we know that we are going to use polar solvent as a mobile phase and the stationary phase is going to be non-polar. So most of our LCMS based methods are like reverse phase chromatography based method. Here there are like two techniques we are using: isocratic mobile phase composition and the gradient mobile phase. Isocratic mobile phase is composed of like example, if you take this example, estranitrile and 10 millimolar ammono acetate, 90 is 10%. So this composition, we are going to use this composition throughout our analysis. But in the gradient composition, there is different change. These compositions, organic versus buffer composition, will be varying throughout the run. So recently, as shown in this picture, I used it for this. I started with isocratic analysis, but I got some interference. This is my peak of interest, but I got some interference from when I used the isocratic method. Then I switched back to the gradient techniques. So like, this is the gradient technique that I use. Run time is 0 to 5 minutes. Flow rate is 6.6 ml per minute. Estranitrile B started with 10% initially, increased it to 90% in 3 minutes. So this is the gradient mode that I used for this drug. So when I used this gradient mode, these two peaks are clearly separated, right? So with I didn't see, when you have the clear separation, that's very good. That's what we need in the mass spec analysis or HPLC analysis. So this is how gradient technique is always recommended, always helpful for to get a clear separation from the endogenous components and to get the highest intensities for our drug. So next, coming to the another important thing is column selection and important impact on the column chromatography. So there are like most widely used column chemistries are here, like C18 columns. C8 columns, phenyl, hexyl, and cyano columns are most widely used in the mass spec analysis. And length starting from 50 millimolar to 150 millimolar millimeters, 50 to 150 millimeters, this one is. And particle size is like 1.5 microns to 2.5 microns. So how do you choose your column? C18 column is like most widely used columns in the mass spectrometry analysis. So because C8, most of the compound, either polar compounds or the non-polar compounds, get eluted in the C18 column. So if your compound is like highly polar, try to use the C8 column because it gives more efficient separation for the polar columns. Suppose if your drug is having phenyl groups or a phenyl hexyl group or cyano groups in their structure. So because these groups are like very highly polar groups, because of this polarity, it is good to use these kind of columns when you develop LC-MS methods. Let's see in this table showing what is the impact of different column parameters in the mass spectrometry analysis or chromatography. So pore volume, it affects the analytical size. Particle diameter, it affects the flow rate. It plays important role in flow rate, pressure, efficiency, and retention time as well. Particle size also, particle type also same. It has greater impact on flow, pressure, efficiency, and retention. Column ID, internal diameter, plays important role in the flow rate. If the column is wider, bigger column, then you can apply a high flow rate. If it is column is shorter, having less diameter, you need to go with the low flow rates. And it has separation efficiency also. Column length plays important role in the pressure, efficiency, and retention time. Stationary phase, that is like what we discussed over here, is C18, C8 properties, different kind of properties. So this plays very important role in the efficiency. So that means separation capacity, retention time, and selectivity. So most widely commercially available columns are like Jorbach's columns, that's from Agilent Technologies, Kinetics column, that's from Phenomenex and Waters VA's column. There are like a lot of commercially available columns other than this available, we can use based on our availability, but it should be same like chemistry wise. If you see, it might be C18 or C8. Coming to the another important thing is like a selection of internal standard. So it is very important to use the internal standard during the LC-MS analysis. So proper internal standard selection is important to minimize your analytical errors. It also minimizes the matrix effect and recovery variations. If it recommended to add the fixed concentration of IS to the each sample in early stage of the sample preparation. FDA and EMA guidelines says that use isotope label compounds as the internal standard. That means deteriorated compounds are isotope label compounds. So what is a deteriorated compound? Here it is. So this is the compound called lorazepam, is the antipsychotic drug. So for this compound, lorazepam D8 is a deteriorated internal standard compound. Here the protons are replaced with a D8 compound, deuterium. So if you see, for this compound analysis, we use this lorazepam D8 as the internal standard. So reason for selecting this internal standard is this internal standard is having similar physicochemical properties like our drug, except the molecular weight changing, that is going to be 8 times more, 8 value more than your parent drug. So it follows same conditions like your parent drug. So for this drug, if you use the C18 column of particular make, and this also will get elute in the same conditions. If you use the particular kind of extraction for this drug, and this will follow your parent compound. Let's see how does it following. So IS selection, here this table displays the data from the non-deteriorated IS. This table I got from using the deteriorated IS. So I prepared 6 different samples and 6 different injections. Let's see how deteriorated internal standard helpful to minimize our analytical errors. In the injection 2, 3, and 5, there is a drop in the drug response, but there is no. I didn't see any drop here in the internal standard response because it's not the deteriorated internal standard. That's why it is behaving differently than our drug behaves. Here, in the same injections, here drug drop response is decreased. At the same time, IS response also decreased. In this injection 2, 3, and 5. Then your area ratio, that means drug area by internal standard area. This is your area ratio. Here area ratio, we always use the area ratio for the quantification, not the area in mass spectrometry analysis. So area ratio is consistent because this one is decreased and internal standard also decreased. You got your same area ratio like earlier. So this is how it minimizes the error. So our acceptance criteria is area ratio should be 0.8 to 1.2 and CV should be less than 15%. So this case it's not within the acceptance limit, and CV also not within the acceptance limit because we use non-deteriorated internal standard. But this case, even though there is a injection variation or response variation, even though you have some sample preparation errors, final outcome is there is no variation because both are decreased, either increased or decreased. That's why you got the acceptance area ratio value within the acceptable limits, and CV also is less than 15%. This is how it is. Our deteriorated internal standards are helpful in our analysis all the time. Coming to linearity range selection, lower limit of quantification, upper limit of quantification, and QC selection. So this is most important step when you develop a bioanalytical method for any kind of drug. Here there are like 2 important situations here. One is like when you know the endogenous levels of your drug. Suppose you are working on one particular A kind of drug. So if you know the Cmax of that particular drug per particular dose, so this is the way to develop a calibration range. Suppose, for example, one drug is having Cmax of 100 nanogram per ml for the dose of 500 mg per mg dose. Then your plan is to construct a 9-point calibration curve. So how to set up the calibration range, highest calibration, that is our standard 9 is highest calibration range. So Cmax into 2 to 3 times. That means you have a Cmax 1000 that multiplies with 2 times. 2000 nanogram per ml is going to be our highest calibration for standard concentration. That's nothing but standard. Once you have this value, then rest of the things is very easy to set up. So this one is 2000 nanogram here, 2000 nanogram per ml is your standard 9. That's, imagine it's 100%. And 80% of this standard 9 is going to be your standard 8. 60% of your standard 9 is going to be standard 7. Then 40%, then 20, 10, 5%. Up to standard 3, you got it like this. Then coming back to standard 1 and 2. So standard 1, always you have a Cmax, right? It is good to take Cmax divides 30 times. So in this case, our Cmax is 1000 nanogram per ml, but divides 30 times is going to be 33.33. That's going to be at least 33.33 is going to be your standard one. That is your lowest limit of quantification limit. It shouldn't be more than that. It should be always less. That's why I took this one as a 30. And 30, that's your standard one. Then standard 2 is going to be 2 times of this one. That's going to be 60. This is how we set up a calibration range. Similarly, quality control samples also we need to select here. So lower limit of quantification, LLOQ-QC is going to be 105% of your standard one. That is LLOQ, right? Up to 105%. I took this one as a 102% here. LQC, lower limit of QC, is 3 times of LLOQ, 30 into 3. It's going to be 90. And the middle quality control is 30 to 50% of your standard 9. Your standard 9 is 2000 nanogram per ml. 50% I took, that's going to be 1000. And high quality control concentration is 75% of, at least 75% of your standard 9. That's going to be 1500 nanogram per ml. This is how we select the linearity range, ULOQ, and upper limit of quantification and lower limit of quantification before you start during the method development. So the one that I explained earlier is when you know the Cmax of a compound. Sometimes we don't know anything about the drug. We just have the drug, we need to develop a method. That time, how to establish a linearity range, that's very tricky here. So in that case, you need to establish a first lower limit of detection. That means how low concentrations you are able to detecting using your LCMS or HPLC, whatever your analytical equipment. Then LOD should be at least 3 times of blank response. Suppose you got blank response of area response of 100. That's on your LOD is going to be 3 times of that 300 area response. Then next, identify the lower limit of quantification. That is nothing but LOQ. LOQ is always at least 5 times of your blank response. Suppose you have the blank response of 100, LOQ is going to be like 500 area. So once you identify the LOD and LOQ values, that's then you can go back to your previous slide. That's how you establish the linearity range, right? You can follow that slides. Suppose how to, so here when I, as I show in this picture, so establishing ULOQ is very important when you here. Suppose I prepared a sample concentration of different concentration, 100, 500, 750, 1000, 1500 like this. I injected into LCMS system. So when I establish the linearity from 100 to 1000, it followed the linearity here. That means when you increase the concentration, response ratio also increase. About the 1000 nanogram per ml concentration, even I increased to 3000, I did not see any response variation corresponding to the concentrations. That means your detector got saturated. So this indicates this is your maximum quantifiable concentration. 1000 nanogram per ml is your maximum quantifiable concentration. Beyond this concentration, it is difficult to quantify your levels. So this is going to be your ULOQ. Now you got the ULOQ. Even if you don't know the endogenous levels Cmax of your drug, once you have this ULOQ, then go back to this table. You can calculate your standard curve like this. So this is how, next slide is like, this is how how to prepare the working solutions here. So, you prepare the stock solution of 1 mg per ml, that is 1000 microgram per ml. From this concentration, these are the yellow ones are the working solutions. Prepare these kind of working solutions initially. From 1000 to prepare 100 to 80, 80 to 60. This kind of dilutions are called serial dilutions. Once we have these kind of dilutions, take these 20 microliters of this concentration, mix with 980 microliters of plasma, that's going to be 1 ml. You got this final concentration in your plasma. So what is your linearity range? 30 to 2000. This is how you got this range in plasma. So once you have these concentrations in plasma, then you can proceed for the extraction setup. How to extraction, how to develop the extraction process and everything. So next, coming to the sample preparation technique. So why we need to prepare the sample for mass spectrometry analysis? There are like 3 important things, reasons we need to prepare the sample preparations. First one is removal of unwanted and matrix component, which causes the interference. Concentration of analyte to improve our limit of detection. Exchange of analyte from our solvent to another solvent, which is compatible with our chromatographic system. Usually, our analytes are present in the plasma or serum or blood. Those we cannot, as I mentioned earlier, we cannot directly inject into the mass spectrometry system. We need to process those samples, extract those samples in suitable organic solvents or polar solvents, then inject into the mass spectrometry. Sorry. These are the most widely used. There are 3 most widely used sample preparation techniques in the during the method development. One is protein precipitation, another one is liquid extraction, third one is solid phase extraction. Protein precipitation used like organic solvents and acids are used for the precipitating plasma proteins. Most widely used organic solvents are acetonitrile, methanol, IPA, acetone. Usually 1:3 or 4 ratios are used. 1 ratio, if you take 1 ratio of the plasma, 3 times of these organic solvents need to be added. Similarly, here acids are also widely used. Trichloroacetic acid, perchloric acid, and metaphosphoric acid are used here. In this case, usually 5 to 10 times are used. Like if you take 100 microliters of plasma, so you need to add 5 to 10 times of trichloric acid to get the protein precipitation experiment. Coming to the liquid extraction, most widely used solvents for this kind of extraction is ethyl acetate, hexane, tertiary butyl methyl ether, dichloromethane, chloroform, diethyl ether, and sometimes combination of these solvents also is helpful. And third and important extraction technique is solid phase extraction. SPE catalysts are used here like HLB, MCX, mixed mode cationic exchange. This is called as mixed mode cationic exchange, mixed mode anion exchange, mixed weak cation exchange, weak anion exchange, and C18 kind of catalysts. Out of these 3 sample preparation techniques, protein precipitation techniques is very simple. You can adopt in our laboratory with minimum equipments. Let's see how does it how to optimize each extraction technique. This is protein precipitation technique. PP is widely used in sample preparation techniques. It's very simple and straightforward. Mostly used for the drugs having more protein binding in nature. If the drug is having more protein binding in nature, more than 50%, this protein preparation technique is most widely useful. So as I shown in the picture, this is how we took the sample. I took the plasma in this tube and mixed. This is the 100 microliters of sample, that is plasma. This sample is mixed with 20 microliters of internal standard. Then I added PP solvent. That's good. That may be like acetonitrile or methanol or solvent or a acid. And vortexer here, like on the vortexer. Once we mix it on the vortexer, then centrifuge that sample for 5 to 15 minutes. Once you centrifuge the sample, what will happen? All solid parts will goes down and clear supernatant, the solvent what you added will comes up. So this organic solvent will crash the proteins. Whatever the drug bind to the proteins will extracted into the organic solvent, whatever we added. So once that organic solvent is separated by centrifugation, we separate that upper layer and take that test analysis into the mass spectrometry. So here the main advantages of the protein, this technique is like simple, very convenient and rapid. We can we need to have only simple vortexer and centrifuge. Once we have these two items, we can do this kind of extraction in our laboratory as well. So, but it has some concerns like possible interference matrix effect. It is difficult to obtain the very clear samples by using this kind of extraction, but widely useful and helpful, cheapest kind of extraction. Coming to the liquid extraction, liquid extraction also widely used in our laboratories for the bioanalytical method development. So this involves, the main principle involved in this kind of extraction is partitioning of sunlight between two immiscible liquids. Our drug always present in the aqueous sample, that is maybe in the plasma, blood, that will be partitioning to the organic solvent, what we are going to use as the extraction solvent. So if you are planning to develop a liquid extraction kind of sample preparation, there are like two important criteria here. We need to think in that way. So most importantly is like sample pH and type of organic solvent that you use for the extraction. So sample pH is very important here, because if the drugs wanted to extract using LLE, it should be ionized or unchanged format. If it is ionization, it won't extract in our organic solvent. To make it unionized, unextracted state, we need to add some buffers or the buffering solution to the plasma or whatever the biological sample you are trying to extract it. So for if it is our compound is acidic or acidic analytes, add acids or bases to make it ionized form. If your compound is basic analyte, basic in nature, add bases or basic buffers to keep it as an ionized form. So this is how we do the liquid extraction. This is the schematic representation of liquid extraction. This is the sample I took in the tube, 100 microliter sample, like protein precipitation, 20 microliters of internal standard added. And I added buffer to make the sample, to keep the sample unionized form. And then I added as extraction solvent here, for example, that could be like ethyl acetate, annexin, tetrabutyl methyl ether. Vortex the sample for 2 to 5 minutes, centrifuge it. And once you centrifuge, you get a clear separation, supernatant organic layer like this. So drug present in the plasma will be extracted into the organic solvent. Take out that organic solvent, evaporate it using nitrogen evaporator, because these organic solvents are non-polar in nature. We cannot directly into the LCMS system. So that's why we are going to evaporate it. The residue again will be reconstituted. That means resuspended in your mobile phase or any of the polar solvent which is compatible, then injected into the mass spectrometer system. So advantages of this kind of LLE extraction is wide range of applicability and clean sample extraction. But this method also having some concerns like labor intensive, it takes a lot of time, larger volume of organic solvent needed, like each sample required around 3 to 5 ml of organic solvent, and then personal health hazard because inhalation of these compounds like a non-polar solvents, right? So inhalation can cause personal hazards. And another kind of extraction technique is solid phase extraction. This is very selective and widely used extraction techniques. It depends on the absorption on the site. Main principle involved here is adsorption. Halides are selectively absorbed on the solid support by the chemical attraction. Interferences are washed out. Then the halides were selectively elated and analyzed here. The most important aspects here is selection of proper cartridge is important. Sample pH is important. Selection of elution solvent is important. As I mentioned here earlier, these are the different kind of cartridges used for SPE sample extraction. So different steps involved in the SPE. There are like mainly 4 different steps involved in the SPE kind of sample preparation techniques. This is the SPE cartridge. It looks like our regular syringe, injection syringe. It has the sorbent bed like this here. So when you purchase this kind of cartridges, these are in a dried condition. So we need to condition it with our organic solvent. Usually, most of the time, organic solvent is methanol. You add about 1 ml of methanol to make it conditionate. Then add 1 ml of water to make it wet. Conditionating in the sense like make it our cartridge is wet condition. Once it is conditionated, next step is loading of your sample. So your sample contain drug and internal standard. That sample mix well and add to this cartridge like this here. Once you add our sample to the cartridge, it passes through the sorbate bed like this. So a red color one is our analyte, and the other are interferences. Our intention is to take out these interferences from our analyte of interest and collect our analyte and use it for the mass spectrometry analysis. So once you pass to this one, then we need to wash. The third step is we need to wash the cartridge with the different organic solvents. So for this step, most widely used solvent is 5% methanol in water, because 5% methanol in water wash out most of the lipids which is present in the biological samples. Not only 5% of methanol, we can use any buffer or 10% of methanol sometimes, depends on our requirement. So most widely used ones are like 5% methanol as per my experience. Once you wash the cartridge with 5% methanol, this interference will go out. So, but our drug always bind to this sorbent only. It won't go out. It won't wash out when you use the 5% methanol. So final step is elution. Elution, in this case, the selection of elution solvent is important here. Most widely using elution solvents are acetonitrile and methanol with some buffer kind of thing. The concept, when you use the 100% acetonitrile, sometimes most of the drugs will elute. If not, use the 100% of methanol. And sometimes we need to use like methanol in combination with some kind of buffers, 1% and 2% formic acid or ammonia kind of solutions are required. So selection of this elution solvent is very important in the mass spectrum, when you develop the solid phase extraction techniques. So this is how your this is how Hello. Yes. Yeah. Sorry. This is how your solid phase extraction equipment look like. This is called as positive pressure 48 solid phase extraction manifold. We can extract 48 samples at a time using this equipment. So now we have optimized our mass spectrometry conditions. As of now, we discussed about the optimization of mass spectrometry conditions, optimization of LC parameters, and the sample preparation techniques of which use your sample preparation and everything. So you have that method ready right now. Before you apply that method to the real-time sample analysis, so we need to validate that method, whether it is suitable for our intended purpose or not. So as for FDA guidelines, these are the different kind of validation parameters we need to perform during the validation. One is selectivity, sensitivity, carryover test, linearity, precision and accuracy, recovery, matrix effect, and different kind of stabilities like bin stop stability, order sampler stability, freeze and thaw, long-term stability, and stock solution stability. Now I am going to briefly discuss. I'm not going to take more time on this because you can read from the bioanalytical method validation FDA guidelines or like EMEA guidelines. I'm going to show some of the slides that I did earlier for the one of the molecule right now. So this is the selectivity results that I got for the one of my experiment. So selectivity is the ability of analytical method to measure the analyte from analyte of interest in the presence of interferences. At least 6 individual lots should be tested. Like if you are developing a method in the plasma, 6 lots to be taken, minimum of 6 lots. This is how it look like. This is the final results I got from the selectivity experiment. So for the lot 1, I got 0 interference. Lot 2 to 1 to 6 also I got 0 interference. So this is the % interference I got for this experiment. So sometimes some of the plasmas are showing interference. Sometimes that's also acceptable, but it should be interference limit should be less than 20%. Sensitivity is nothing but establishing your lower limit of quantification, LMOQ. For this compound, I have established like a sensitivity of 0.4 nanogram per ml. So I injected at this concentration 6 samples. Then I injected 6 samples. This is how I got it. CV is 14%, accuracy is 111%. Acceptance criteria is like less than CV should be less than 20%, and this accuracy that also called percent nominal is 80 to 120%. So our results are within the acceptable limits here. Third one is the carryover test, injector carryover. Sometimes when you inject to the higher concentrations in the mass spectrometry system or HPLC, there might be possibility of getting the injector carryover or carryover from the column as well. So this is how we injector carryover experiment we have to done. So inject 2 blanks initially, then inject the highest concentration, like that could be your standard 9 or standard 8, whatever the highest concentration. Inject that sample, then re-inject these 2 blanks. So initially, then check for the interference. In this case, after injecting the highest concentration, I did not see any interference. That means percent carryover is 0. Sometimes there might be chances of getting the carryover. If you get any carryover after injecting the highest concentration, that should be 20%, less than 20% when you compare with mean LLOQ value. Then only your method is shown to be no carryover effect. If this interference is more than 20%, you need to optimize your rinsing cycle or rinsing step, rinsing solution in the that's going to use in the HPLC. Linearity. Coming to the linearity means establishing a calibration curve. This is the linearity range that I selected for the compound, 0.2 to 10 nanogram per ml. At least 3 linearity curve needed for the during the validation, but I did 4 experiment here, 4 curves here. On the slope, always correlation coefficient should be 0.98 and greater than for each of the curve. Next one is precision and accuracy. So here during the validation, we need to conduct at least 3 precision and accuracy batches. Each batch contains one blank sample with internal standard, one calibration curve, that means standard 1 to 8 or 9, whatever you like, but minimum we should have 6 standard points in the calibration curve. Above, you can use either 9, 7, 8, 9, 10, that's okay. Minimum we should have 6. And 6 set of quality control sample. That means each of 6 one. This one LLOQ6, LQC6, MQC6, and HQC6. And as I said, 3 precision and accuracy batches we need to perform during the validation in 2 different days. One is 1 day, and next 2 are another day. So from the first day experiment, calculate the intraday precision and accuracy, and next 2 batch all together intraday precision and accuracies. This is the values I got from the experiment, CV and accuracy here. So like I mentioned earlier, acceptance criteria for CV is less than 15%, but LLOQ case, it is less than 20%. Percent accuracy, this value should be 85 to 115% is, except for the LLOQ, which is 80 to 120%. Recovery is determining the extraction efficiency, your extraction method, how efficient it is. So recovery should be conducted at 3 concentration levels, that is low, middle, high quality control concentration levels. Inject 6 samples of extracted sample and then neat samples like this. Neat samples are prepared in just the solution, not in the plasma or any of the biological samples, biological matrix. Like this, extract 3 concentration levels. So you got the areas of 6 injections, mean of this 6 injection, mean of this one. So extracted area by neat area, that gives our recovery value at this concentration level. That in my case is like 76% at LQC, 71% MQC, and 76% is HQC. So overall mean of these 3 recovery values is going to be mean of your analyte recovery. That is going to be 74%. There is no limit for the recovery. It can be any value up to 120%. But it should just even 10% is also okay, as long as you are able to quantify the drug in the using your method. And final important experiments here is like stability, benchtop stability, order sampler stability, freeze-thaw stability, long-term stability. Benchtop stability is nothing but establishing the stability of your drug or your analyte in biological samples at room temperature. BTS should be established at least for 4 hours. So spike your drug into the plasma or whatever the biological sample, keep it for at least 4 hours in the room temperature, and check for the stability. That's called benchtop stability. Order sampler stability. Next one is order. Once you process, you already established one extraction procedure during the method development. Using that extraction techniques, process the samples, keep it in the order sampler, wait for 24 hours. After 24 hours, inject those samples. After 24 hours, inject those samples, then check for the stability. Third one is freeze-thaw stability. So freeze-thaw stability means all plasma samples or any of the biological samples, we are going to store at -20 or -70 degrees temperature to keep it store it for longer period of time. Sometimes we do the analysis couple of times or 3-4 times for each sample. So the intention of this stability, freeze-thaw stability, is to even repeated freezing and thawing of particular sample does not compromise your stability. So you have to establish at least 3 freeze-thaw cycles, which with the sample stored at -70 degrees centigrade during the validation. And most importantly, another important stability is long-term stability. So long-term LTS, called as LTS, it is long-term stability is analyte measuring the analyte stability in the biological samples stored at intended temperature for a period of time. LTS should be established based on the how many days of real-time samples are stored. Suppose your clinical sample or preclinical samples are stored for 3 months. You need to establish long-term stability for about 3 months. So all stability experiments should be conducted at LQC and HQC concentration level. For each concentration level, we need to prepare 6 samples, n equals 6 that is. And all these stability sample must be compared and quantified using the freshly prepared calibration curve and quality control samples. This is one of the example of the data that we generated from the recent stability experiment. So as I mentioned, all stability should be conducted at LQC and HQC at 6 levels. This is the concentration of LLVQ, nominal concentration. That means this is the concentration I prepared in the plasma. These are the concentrations I got for the after the different stability. Like suppose if it is a best of stability, this one I got it. These are the stability. That means this value by this value into 100. That means calculated value divides nominal concentration multiplied with 100 is going to be our accuracy. Similarly for the HKC also here. So overall stability is here, mean value, this one. This mean value divides with nominal concentration multiplies with 100 is going to be stability. All the time, for all the stabilities, CV should be less than 15% and stability should be 85 to 115% during entire course of validation. So you developed a method, you are validated. What is the next step here? Next step is analyze your samples. What is your purpose? Maybe you are going to analyze like preclinical or clinical sample, whatever the sample you are going to have it. The next step is you can analyze, method is ready for the analysis. So this is, I'm going to wrap it up my presentation right now. So I would like to express my deepest thanks to Dr. M. Vijayalakshmi Madam, Associate Professor and Organizing Secretary, for giving me this opportunity. Also, I would like to express my thanks to Dr. Sunita Madam, Professor and Principal, and KVSR Siddhartha College of Pharmaceutical Sciences Management and staff for this opportunity. Also, thanks to audience. I'm happy to take any questions if you have it.
Thank you, sir. Thank you very much. A great talk on bioanalysis. Now, I request Rihanna ma'am to just go ahead with the queries.
Hello, good morning, sir. Myself,
Good morning.
Assistant Professor from KVSR Siddhartha College of Pharmaceutical Sciences. Sir, here our participants have some questions, sir. First
Yes,
one,
ma'am.
if we want to isolate analysis from biological fluids like blood, urine samples, so can you explain any one example how to isolate or how to prepare any one example, sir, from biological fluids?
Analgesics.
So, analgesics.
Analgesics.
Oh, okay. I'm thinking your voice is a little bit low, madam. Sorry about that. Okay. Any analgesic compound, right?
Yes, sir.
Analgesics are like we have diclofenac,
Yeah, aspirin, paracetamol.
aspirin, paracetamol, ibuprofen, mefenamic acid.
Yes, sir. Probably we have this topic in our MPharm PG syllabus.
Okay.
So how
Okay,
to
ma'am.
isolate as per our lab structure.
Yes, madam. So you get for irrespective of the category, the procedure is same for the most of the compounds. Like we need to as per the facilities that you have in our laboratory, I would suggest to use the protein preparation techniques, which is very easy. You just need just the vortexer and the centrifuge for those kind of experiments. So spike those, whatever the drug sample you have in the plasma, crash it with estronitrile or methanol, centrifuge it, separate the supernatant clear liquid and use it, that liquid for, inject that liquid for in your HPLC. It's like a very straightforward, nothing related to the category of the drug. And also you can use liquid extraction techniques, because analgesics are like most widely used drugs. You can use for the extraction to make a clear liquid. LLE also helpful. More in the LLE, tertiary butyl methyl is most widely used in solvent. TBME we call it, right? So that you can use, or ethyl acetate sometimes. And diethyl are also very good. It's like environmental friendly organic solvent. You can use that for the separation of these other analysis kind of related things. And coming to the LC part, most of the analytics are separated using C18 columns. You can use any C18 columns that was available in your laboratory. That could be maybe different. It doesn't matter what is the make of that column. The chemistry is important, as I mentioned. C18 column is helpful for in your case. C18 column, liquid extraction or protein presentation extraction will helpful in this regards.
Okay, sir. Thank you. And next one is what are the precautions to be taken when we need to introduce biological samples in HPLC column? Like now you told us, sir, C18 column.
So you are asking, yes, madam.
What are the precautions we have to take when we insert the biological samples into the HPLC column?
Most importantly, sample preparation. So when you always, our aim is to get the clear. Always our intention is to get the clear sample. So here the precaution is, once you have the clear sample, it is nothing, and it won't harm to your HPLC system. So one more precaution you need to take, once you use the HPLC system, means you have analyzed 20 samples today. After completion of your analysis, wash this column with at least 80% of your organic solvent. Initially 50-50 water and organic solvent, any of the, suppose you are using estronitrile as a mobile phase. So when you, at the end of the day, once you finish your experiment, wash your column with estronitrile water 50:50 ratio for about 1 hour, and then wash the column with 90% or 100% of organic solvent for about 30 minutes. That will helpful. That will give more life to the column as well as our HPLC also.
Okay, sir. The next query is, how can we prepare protocol for the bioanalytical samples method development? For bioanalytical method development, according to US FDA guidelines, how can we prepare protocols?
Here, can I show you one protocol model?
Yes, sir. Can we provide any one example?
Yes, ma'am. Give me a second, I'm just opening that my report. Can you able to see my screen again?
Yes, sir.
Yes, ma'am. So this is a report or a protocol or a report, bioanalytical report look like. This is the title page. suppose it could be sometimes method validation protocol or method validation report. Both are same almost. So this is the title of that report: validation of high performance liquid chromatography, mass spectrometry, method in positive mode, estimation of colchicine. Colchicine also is like a gout drug, I guess it is. I'm not sure. In human plasma using colchicine D6. This is the report number. What is the report number, like versions and everything when you drafted this report? That means when you guys signed on this report, that is the final date. Then this is the content of the report, table content. And then list of figures, list of abbreviations, and summary of the report. Before that, there should be some signature page. So who is the that signature page says that who is the bioanalyst, who is the reviewer, who is the authorizer, who approved it is. So here in this report, that page is missing. And other than this, everything, this is the summary of the method used. Objective, what is your objective? What are the experimental conditions used here? What are the reagent, batch numbers, manufacturers, and everything? And working solution, working standards, colchicine, what is the purity, where you get this one? What is the retest date or expiration date? These are the solutions, how you prepare the solution preparation for this experiment. And what are the biological matrix? What kind of plasma usually here and everything. So how you prepare the calibration study standards and quality control samples, this one. And what are the mobile phases used? And bioanalytical condition, column, mobile phase, flow rate, injection volume, LCMS conditions, what is the parent byproduct mass spectrometries, what are the different gases and potentials, what kind of sample preparation used. And coming to final, fourth section is results. What are the different validation parameter results? Finally, it's with tables. What are the result tables? What are the results you got from the analysis? This is going to be like around 50 plus pages. At the end, chromatograms you generated during the analysis.
Okay.
So, is it clear, ma'am?
Yes, sir. Yes, sir.
Thank you.
Sir, can you show us any protocol for bioanalytical and bioequivalent studies according to US FDA guidelines?
Okay, bioequivalence study. Let me check if I have it or not. Bioequivalence. Not sure. See, 1 minute, madam. I'm trying to find out the report because I'm not yet ready with this report. Sorry about that.
Yes, sir. No issue, sir. You can send us later so that we will send to us.
Yeah, definitely, madam. I will find out it. I can share you whatever you want. I will give my email ID. You can send me emails and everything. I can share it to you. Definitely. Sorry about that. I need to check where it is.
Okay, sir. It's okay, sir. Another query from one of the participants, sir. So, on what basis we select the internal standard and the concentration level?
Yes, that's a good question, really. So, I'll tell you with the example. 1 second. So question is that at how you select the internal standard concentration, right?
Yes, sir. Internal
Yes,
standard.
that's good.
How we select
Internal standard concentration. Yes, I got it. So first, so here, when you choose the linearity range, middle QC concentration is 1000, right? So when you have this concentration, when you inject into the mass spectrometry for the 1000 nanogram per ml, suppose you got the area of 10,000 or 1 lakh, whatever area for 1000 concentration. This is for the drug. For the internal standard concentration also, you need to select the concentration that gives equivalent or similarly closer area to the MQC concentration. That means MQC of analyte giving 1 lakh 1 lakh area counts. Your internal standard also should give 1 lakh area count. Suppose your internal standard giving 1 lakh area count at a concentration of 100 nanograms. That's going to be your internal standard concentration. You understand my question, or can I explain it like once again? So that the area of internal standard, when you are getting, should be matched with the MQC concentration area. Similarly, whatever the MQC concentration, it is good to use internal standard concentration similar to middle quality control sample concentration. Ideally.
Okay, sir. Sir. Another query, sir, from one
Yes,
of the
ma'am.
participants. So why bioanalytical works are not preferable to publish in high impacted journals?
Why not? High impact factor means which journal, I'm not sure. But because as per my experience, my work is published in bioanalytical work I'm talking. Impact factor of around 6.
Oh, great, sir.
6 is good enough, right?
Yes, sir.
So the question is really good. Yeah, might be because bioanalytical is always like a supporting. It's not the. Most of the bioanalytical work is supporting based work. We support to the formulation scientist. We support to the clinical scientist. We support to the other kind of scientist, right? So we are not directly discovering any new drug kind of innovations here. It's a support kind based work. That's why it is difficult to publish directly LCMS methods in the like in JAMA or those kind of journals, which is having 20, 30 impact factors. When you have the real discovery or real innovation kind of thing, then only we can publish in those kind of journals. But our journals also good, like Journal of Pharmaceutical Analysis is having very good impact factor. Journal of Chromatography B is having around 3.5, Chromatography A is having 5 impact factor. Analytical chemistry is having 5 impact factor. 5 impact factor is really good. But doesn't matter whether the impact factor is more or less, but publishing data is important in my experience.
Okay, sir. Another question is it
Yes.
possible to detect impurities by HPLC?
Yes, it is. Yes, it is. When you HPLC is always inferiority profiling will be doing in the HPLC most of the time. So if impurities are really in the picograms and nanograms level, it is difficult. Most of the impurities are in microgram level formulation. It's present in the formulation, right? It's easy to detect in the HPLC. We can do that.
Okay, sir. Thank you for your valuable informative session. So now Ma'am,
Yeah,
Vijayalakshmi
thank you,
ma'am.
sir. Yes, sir. Come on. Thank you, sir. And one more question. Like, is it essential while we go with liquid extraction, this nitrogen evaporator you have given us, sir, in the last step, to evaporate that solvents in liquid extraction?
Yes, madam.
So alternative for that nitrogen evaporator.
Oh, alternatives are like, yeah, that is very important, madam. Nitrogen evaporator here. Alternatives are there. Vacuum evaporators are there. That's also, I think it's a similar costly, equivalent cost to the nitrogen evaporator maybe. But I don't think so. Nitrogen evaporators are too expensive. There are like cheapest ones are available that we can afford in the small laboratories as well. But nitrogen evaporators are very good. But without nitrogen also there are like vacuum evaporators. Those are called vacuum evaporators. But if you want, I can send you some of the links later.
Sure, sir. Sure, sir. Thank you so much. Thank you very much. Sure. Like you told you about various makes, models, softwares of LCMS in your presentation. And that
Thanks,
is
ma'am.
about the principle and instrumentation in brief. You told that we are going to cover about the optimizing conditions. Still, you have touched that ESI, AFPCI techniques, what it is for actually, and about the positive, negative modes, what type of samples we are going to achieve in positive and negative modes, and about the optimization, very clear and clean explanation, I can say. Like about the buffers, isotopic, whether with the examples you have shown you about the solvents even here. Because generally without basic chemistry knowledge, we cannot go with the different types of solvents choosily, right? And moreover, academics, we are going with particular solvents like methanol, nitrite. Only few solvents we are going to choose generally. So maybe that might be the problem sometimes without having an awareness of solvents, we'll go with these two types. And you have told about the internal standard selection clearly in the question, whatever you have, and about the validation, even you touched that LLOQ, which is actually a confusion matter for academic students. When they enter into the industry, obviously they'll be knowing about how to fix this LOQ and LOD values based on Cmax. So generally we won't use this word Cmax and all while we develop a HPLC method. Generally based on the sample properties and all, or else the pKa value, or else trial and error method will go. And so that's a broad explanation for us about sample preparation along with solvents. So from now onwards, we'll try with this protein precipitation liquid extraction very easily, I can say. But of course, here I want to ask one more question, sir, about the column dedication. Generally, in industries, they used to dedicate the columns for particular samples, right? So whenever we go with different types of samples, so is it possible, it is not possible for us to dedicate the column actually. So most of the drug samples, and of course it depends upon the drug availability also, as you know. So based on that, we are going to choose the same column for all the types of drug molecules.
It is not required, but it is not required to dedicate the column. You can use any of the, so suppose same column used for one kind of drug analysis today, the same column you can use tomorrow, you can use the same column for the another drug as well. Only thing is that when you finish the column analysis today, make sure to wash your column with, as I mentioned, with initially with the water resveratrol 50% or water methanol 50%. And that's for half an hour at least. And then 80 to 90% of organic solvent, whatever you are using in the mobile phase, it is better to match with that. So without buffer, madam, suppose your mobile phase is acetonitrile and ammonium formate buffer, then your washing solvent should be acetonitrile and water for 30 minutes, 50:50 ratio, and another 30 minutes for acetonitrile alone.
Is that
That will
okay,
helpful to you.
is that okay for sending this 100% organic solvent towards the column?
That's fine, madam. You can send it. But if you have any concern with this, just use 95%, 90% to 95%, 5%. It's not hurt the column. Sometimes, as you mentioned, maybe chances are there. For safer side, use 90 to 95% of organic solvent, 5% of water. That's it.
Thank you, sir. Thank you very much. And I found that most of the research scholars, while feeling difficult in preparing this sample in their labs from biological fluids, whereas actually for PhD thesis and all, we need to submit the sample preparation from biological fluids only. By having this three-dimensional information like spectrum, we need to present in our thesis. Otherwise, it is not valuable actually. And with that only, we'll be able to get papers also, articles also. So we have so much of information from you, sir. Thank you very much, sir.
Thank you, ma'am. So if anyone needed any help in this regard, or later if you guys have any questions, you can reach out to me. Feel freely always email me, or so I will share you this presentation to you later, madam, tomorrow. Whenever you have any doubts, or you can reach, you can refer this presentation, or directly email me.
Okay.
Sir, another query from one of the participants.
Yes, madam.
Hello.
Yes, madam. I'm there.
How do we select the dose for animals during bioanalytical method development?
Those for animals is a part of the DMPK scientists. They will decide. We are not going to select the dose here, but if you wanted to do the dosing in our laboratory, I'm not particularly sure about how do you choose because I'm not a DMPK scientist actually, because I don't do the dosing and everything here. I can refer something and answer this question later, because I never do the dosing. I am being a bioanalytical scientist or analytical scientist. We don't do the dosings. DMPK people, like those who work with the animal studies and the preclinical trials, they do the dosing, collect the sample, give it to us, we analyze it. I believe that dosing will be depends on the type of the drug, like how it matches to the human dose. Suppose one drug we are using in humans like 500 mg per ml 3 times a day, or like ODR 2 times a day, right? So this will be similar dose will be used to the animals also. Per kg weight, that should be for kg 500 mg for human dose. If it is a 500 mg for mg tablet we are giving for 3 times a day, calculate it as a weight of the human. So how much is, suppose average weight is like 50 to 60 kgs, right? 500 mg we are giving to the 50 to 60 kgs of human being. The same weight animal, same like this, calculate the weight of the animal. Based on that, back calculate the how much we can give it to the animal. Maybe like this, but I am not exactly sure of this one. But I can refer for you later.
Okay, sir. Thank you, sir.
Thank you, sir. Thank you very much. And on behalf of our principal ma'am also, I'd like to thank. And one more thing, I can request you, like whenever you come to India, please come to Vijayawada.
Sorry,
And let us privileged. And thank you very much, sir.
Thank you.
For the participants, you please fill the Google form, whatever you have given the WhatsApp group for your attendance. Thank you very much. And sir, probably in the future, I mean to say for our informed practicals or the research scholars, if we have any queries, we can give your mail ID to all our students, right?
Sure, madam. Anytime mail ID, and I can give my phone number also. That's not a problem. This is my phone number. 1 minute, madam.
sir. Thank you very much, sir. Thank you once again for your.
So if you have any questions, if you need any help, use my WhatsApp number. You can text me or call me. Just inform that I'm from Siddhartha College to get to know.
Sure, sure, sir.
That's
Thank
not a
you very
problem.
much, sir, once again.
No problem.
We can be the two, sir.